We're still looking at some of the basics of bacteriology to help to give you a better understanding once you start doing the individual genera and the individual species. And so today we look at antibacterial, antimicrobial agents. And this will be the outline of our presentation, a bit of introduction. So this is not a pharmacology lecture, and so we will not go into the details of the pharmacodynamics and the pharmacokinetics of the antibacterial agents that we'll be looking at. So this is looking at antimicrobial agents from the point of view of bacteriology, with a foundation of having looked at the bacterial cell structure, because the bacterial cell structure is critical in understanding the targets for the antibacterial agents. Then we'll classify the antimicrobial agents. Again, from the point of view of bacteriology, you may use a different classification when you're looking at when you're in pharmacology. Then we'll briefly outline the antibacterial agents. So it will be a mention. You will do the details, as I said, when you're doing the pharmacology. Antimicrobial resistance, increasing problem worldwide. So we are going to mention antimicrobial resistance. There's a lot of reading in antimicrobial resistance, so I will introduce it and leave it to you to go and read more about the antimicrobial resistance, what research is being done about it, why we're worried about it. And finally, we are going to look at what you'll be looking at in the lab, antimicrobial susceptibility testing. So this will be the practical introduction to what you will do in the lab. So as you look at antimicrobial agents or antibiotics, there are some definitions that are important to understand from the outset. Some are used widely, but probably wrongly. So it's important to understand the meaning of these terms. So the first one is an antibiotic. So you see, from the point of view of bacteriology, an antibiotic is a chemical substance which is produced by various species of microorganisms. So it's a natural product. Important to remember that is capable in small concentrations of inhibiting the growth of other microorganisms. So scientifically, when we talk about antibiotics, we are very specific to that chemical substance which is naturally produced by some microorganisms. There may be bacteria, of course, you know the story of penicillin, and that it was accidentally discovered when penicillium, the fungus penicillium, was grown into a petri dish, or grew. It was actually a contamination into a petri dish containing a bacterium known as Staphylococcus aureus. So that's an antibiotic. So with time, it was realized that bacteria get resistant to these antibiotics, and so there was need to improve the molecules. So that's why we have semi-synthetic antibiotics. So the original was antibiotic, but then there was an addition of some group, probably to give it a special characteristic. So these are synthetic derivatives of naturally occurring antibiotics. And then we have the chemotherapeutic agents, which in bacteriology we refer to, or we define as chemical antimicrobial agents. So these are purely synthetic agents. Then we have the term that we prefer to use in bacteriology, which is an antimicrobial agent. And this is because it encompasses everything that the three definitions that we've had earlier. So antibiotic, synthetic antibiotic, chemotherapeutic agents. And remember, you're looking at low concentrations. So because remember, you have a patient that you're giving the antimicrobial agent to. So you do not want side effects. The higher the concentration, the higher the risk of side effects to the patient. So they inhibit the growth of other microorganisms. So specifically for the antibacterial agent, there are again classified into two, depending on how they perform their role. So either bacteriostatic agents or bactericidal agents. So bacteriostatic agents are antimicrobial agents that inhibit the growth and the replication of bacteria. So they do not cause the death of the bacteria. So they're non-lethal. And then we have the bactericidal agents, which cause bacterial cell death. So they could do this either by inhibition of cell wall synthesis, or nucleic acid synthesis, or protein synthesis. So let's see how much you remember about the cell wall. What's the difference between the gram-negative and the gram-positive cell wall, bacterial cell wall? So you can either unmute or write in the chat. What's the differences between the cell wall, gram-positives and gram-negatives?
Gram-positive has a thicker cell wall than the gram-negative.
So NM says negative have a thicker wall of what is a proteoglycan? Is that correct? That's incorrect. So the peptidoglycan is thicker in the gram-positives. Gram-positive has a thick peptidoglycan layer and a thin peptidoglycan layer. So when you're writing your essays, do not write these abbreviations. So make sure you write out the entire term, peptidoglycan, not PG. So positive has a thicker layer of peptidoglycan above the plasma membrane. So what do gram-negatives have? What layer do gram-negatives have that gram-positives do not have? I was also looking at that. Yes, that's correct. So gram-negatives have an outer membrane. Outer membrane, not surface membrane. The outer membrane, how do molecules get through the outer membrane of the gram-negatives? So remember, the peptidoglycan layer is freely permeable, but the outer membrane of the gram-negatives is not freely permeable. So that's correct. Abdallah, just current through porins. What are porins? So these are protein channels that are not freely permeable. So you need to remember the weight and the diameter of the molecules that they get through. That will be important when you're looking at one class of antibacterial agents. There are some antibacterial agents that you cannot use in gram-negatives because the molecule is too large to pass through the porins, which is why it's important to understand the bacterial structure before you can discuss the antimicrobial agents. So another term, sorry, which is important in antibacterial agents, or indeed any agent acting against an organism, is what we refer to as selective toxicity. So selective toxicity is the attribute which the ideal antimicrobial agent would have. We do not yet have the ideal antimicrobial agents, but when you're looking later on in this course at the antifungal agents, there are some antifungal agents that come very close. So what is selective toxicity? So you only want to select the target to be on the organism, but the target should not be on the host, because most of the side effects arise from the fact that you have the target in both the bacterium, in this case, and also the human cell. hence the side effects. So if you had an ideal antibacterial agent, it will be perfectly selective, selectively toxic, which means that it's only targeting the bacterium and not targeting any part of the human cell. But we still haven't got the ideal antimicrobial agent. So in summary, selective toxicity, highly effective against the microbe, but have minimal or no toxicity to humans. So we do not yet have the ideal antimicrobial agents. But of course, efforts are being put to ensure more selectively toxic antimicrobial agents. So how do we classify antimicrobial agents? So two broad ways of classification. Remember that we are looking at it from the point of view of the bacterium and not pharmacology. So you may come across different classifications in pharmacology. So here we are interested in the targeted microorganism, and on that microorganism, what's the mode of action? So you can have antibacterial agents, and you also have some classifications. For example, the antimicrobacterial agents, which are specifically, or they are reserved drugs that target bacterium in the genus Mycobacterium. Why? Because we have important infectious agents like Mycobacterium tuberculosis, which causes TB. So we have reserved drugs specifically for the Mycobacterium. They can be used against other bacteria, but it is best to reserve them to avoid overuse, and overuse, as we shall see later, causes the risk of resistance. Then you have antifungal agents, antiparasitic agents, antiviral agents, which I am sure you've already done. And then the mode of action. So the mode of action in bacteria, which is what you're going to look at. So we are going to look at antibacterial agents, and the mode of action is classified according to the target in the bacterial cell, which is why it is important to first understand the bacterial cell structure before you can look at the antibacterial agents. So if there are any questions as we go along, you can either unmute or write in the chat. So the first mode of action is interference with cell wall synthesis, which is why we went through a summary of the cell wall. So remember, gram-positive bacterial cell wall, gram-negative bacterial cell wall. So they interfere with cell wall synthesis. Now, the cell wall biosynthesis, the bacterial cell wall biosynthesis is a complex activity. We shall not go into the details. For those who are interested in getting deeper, there are excellent articles that you can look at about the cell wall biosynthesis. But what you need to remember at your level, as we said, is that the two basics, NAG and NAM. What does NAG stand for? NAG?
N-acetylglucosamine.
Let's see if you remember. So NAG, N-acetylglucosamine, 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 yeah, N-acetylglucosamine. What about NAM? What does it stand for? Yes, NAM is N-acetylmuramic acid. And remember, we said that NAM carries a short peptide. We have the generic short peptide, but sometimes it changes depending on the bacterium. But generally, you have L-alanine, D-alanine. So remember what it stands for. We had covered this when we were looking at the cell wall. And what makes the bacterial cell wall so strong? Remember, we said it's the interlinkage. You can see the peptide chains to your right. So you have NAG-NAM, NAG-NAM, you have the short peptide, and it's through the short peptide, the interlinkage of the short peptide to the other chain of NAG and NAM that makes the cell wall so strong. And remember, for the gram positives, you have the teichoic acids, you have the lipoteichoic acids, and so on and so forth. So the first group of antibacterial agents that interfere with cell wall synthesis are the beta-lactam antimicrobial agents. And these are by far the agents that you're going to be dealing with in your practice. So the beta-lactam antimicrobial agents feature a beta-lactam ring in their structure, hence the name. And this beta-lactam ring mimics the D-alanine moiety, and they act on the formation of the peptidoglycan layer. So when you look at the interlinkage, you can see that the linkage usually happens the D-alanyl from one chain to the other chain. So the D-alanyl, D-alanine linkage. And so the beta-lactam ring will mimic that ring. So a bit of background is that the building of the chains that we've been talking about and the cross-linkages in the peptidoglycan layer are catalyzed by specific enzymes. And these enzymes are members of a very large family of enzymes referred to as the serine proteases. Now, these enzymes are also referred to as the penicillin binding proteins. And this is because they target the beta-lactam antibiotics. Penicillin, by now you should know, is the largest group, or was the first molecule to be discovered in the beta-lactam antimicrobial agents. So the commoner name that we use for these enzymes are the penicillin binding proteins. So how do they act? They form an irreversible intermediate within the active site of the penicillin binding proteins. And then this weakens the peptidoglycan layer because you have stopped the formation of chains, you have stopped the interlinkage of these chains. So this will weaken the peptidoglycan layer and the subsequent inhibition of cell growth. Remember, we said the cell wall is very important for cell growth. And because of this, then the cell wall is not able to maintain the internal osmotic pressure, and there is subsequent lysis. And of course, with lysis means that you have cell death. And so beta-lactam antimicrobial agents are bactericidal because they result to the death of the cell. So again, we'll not go into the details. You're going to look at the details in pharmacology. But just to mention them, we have the large group of the penicillins, and there are different generations of this, as you shall be told in pharmacology. And penicillins are antibiotics, so because they're natural products of the fungus Penicillium chrysogenum. And then we have the other large group, the cephalosporins, which are also in different generations. So these are also antibiotics derived from the fungus Cephalosporium. And in this case, the beta-lactam ring is fused with the dihydrothiazine ring. Again, the details will be clearer when you do pharmacology. We have the cephomycins, which contain oxygen in place of sulfur in the dihydrothiazine ring, and more stable to the beta-lactamase hydrolysis. We shall talk about the beta-lactamase in a few minutes. Then you have the monobactams. Now, these are narrow spectrum. So antimicrobial agents can either be broad spectrum or narrow spectrum. So broad spectrum antimicrobial agents can are able to target a wide range of bacteria, while narrow spectrum antimicrobial agents, as the name suggests, have a narrow target. So the monobactams have a narrow spectrum. They target aerobic gram-negative bacteria. An example is the bacterium Pseudomonas aeruginosa, which you're going to cover. Very important in because it shows high rates of antimicrobial resistance. And an example of a monobactam that's widely used is Astionum. Then you have the carbapenems, which are broad spectrum. An example is imipenem, meropenem, itapenem, and doripenem. So someone is asking to repeat the mechanism of action of the beta-lactam antimicrobial agents. So to understand it, remember, we always go back to the basic building blocks of the peptidoglycan layer. And specifically for the penicillin binding protein, specifically in the cross-linkage. So remember, we have the short peptide that's carried on NAM, and then you have the transpeptidation or the cross-linkage, the L, the alanine, D-alanine. So that's where the cross-linkage happens. So this process is catalyzed by the large group of enzymes that refer to as serine proteases. But usually when you're talking about the beta-lactams, we refer to them as a penicillin binding protein. So these are enzymes that catalyze cross-linkage of the different chains. Now you have the beta-lactam antimicrobial agents. You can go and look at the different structures, compare the structure of the beta-lactam ring with the D-alanyl D-alanine. So what happens is that the beta-lactams will attach themselves to the penicillin binding protein, mimicking the D-alanyl D-alanine. So you need that cross-linkage for the strength of the peptidoglycan ring and for the continuation of the formation of the peptidoglycan layer. So obviously the beta-lactam ring is a dead end because it cannot continue. And that is what causes the weakening of the peptidoglycan ring and the subsequent lysis, because they are not able, the cell wall is no longer able to maintain the high internal osmotic pressure. So I hope that's clearer. It's a whole process which, if you're interested in the details, you can look for it. You can Google. There's some good papers that talk about the action of the beta-lactam antimicrobial agents. So for here, we are doing the introduction. We'll not go into the details. Okay, so it's clearer. And I think this one will also make it clearer. So you can see the rings, the cross-linkage to your right. Is it your right? I should be your right because you're on a screen. So the cross-linkage, the transpeptidation, the D-alanyl D-alanine, that's the basic, but you can have some changes. Sometimes bacteria change these, which is part of the antimicrobial resistance. And so what the beta-lactams will do is to attach the peptidoglycan into the penicillin binding proteins. So that will be a dead end because no more, you cannot continue with the process of cross-linkage and subsequent lysis. So at the bottom here, you have penicillin ring, and then you have the D-alanyl D-alanine ring. So you can see some changes which will cause the stoppage of the formation of the peptidoglycan layer. Now to your left, you have the basic structure of penicillin and cephalosporin with what we refer to as the R groups for variations. So a bit of history, I do not know if I have that. So a bit of history, of course, there was a lot of excitement once penicillin was discovered. The scientists assumed that they had found the wonder drug. But very soon they realized that bacteria were producing enzymes that were breaking open the penicillin ring. Okay. And once the penicillin ring is open, it can no longer be effective against bacteria. And that's why you have different generations of penicillins and cephalosporins because of remember the synthetic antibiotics. Sometimes you need to add, what is the word? You need to add specific advantage to penicillin. We shall look at it in the next one. So you need to add a specific group which will make it better able to handle the enzymes that are produced by bacteria to cleave or to open up the beta-lactam ring. So hence the R groups. So when you're looking at the different generations of penicillin, I'm sure in pharmacology, you'll be looking at the chemical structure. So you'll see for the later generation cephalosporins, they carry their own beta-lactamase inhibitors, as we shall see later. So let's explain what beta-lactamase is. So you should have understood that the beta-lactam ring mimics the D-alanine ring. I hope you've understood at least very basically the process that's used to target the bacteria and to stop the formation of the cell wall. Now, what was realized is that bacteria produce enzymes that break open or that cleave the beta-lactam ring. And this large group of enzymes are referred to as the beta-lactamases. So what are beta-lactamases? Now, again, these are serine proteases. Remember, we said serine proteases is a large group of enzymes that have different targets that inactivate the beta-lactam antimicrobial agents. Now, there's a whole other very large and detailed topic that if you're interested, you can look at. It's not necessary, but if you like details, you can read about around the beta-lactamases. So they inactivate the beta-lactam antimicrobial agents. Different ways of classifying, but the most basic is it narrow spectrum or is it broad spectrum? So the narrow spectrum have a specific target. For example, the penicillinases, as the name suggests, will only target the penicillins. You have the cephalosporinases, which will target the cephalosporins. You have the carbapenems, which will target the carbapenemases. And then, of course, you have the broad spectrum, which you really worry about, because here you have a beta-lactamase, which is targeting all the antimicrobial agents that you're giving to the patient. Another way of classifying is using the class A, B, C, D, sometimes because of molecular studies, and you're better able to study the structure of these beta-lactamases. Some of these classifications may not be as recent. So you can look and see, is there class E? Is there classification for class A? So this was a class of 2021. So class A, the beta-lactamases. So you have SHV1 and TEM1. So there's a point mutation which produces extended spectrum beta-lactamases. Now, cephalosporins, although I didn't want to go into the details of the classification of the cephalosporins, you have the extended spectrum cephalosporins. So you have, I think that's group 3 or 4. So you have the basic cephalosporins. So what was realized very quickly is that bacteria producing beta-lactamases and saw groups. So you saw the R groups on the sides of the molecular structure. So there were groups that were added so that for the later generation cephalosporins and penicillins, they carry their own beta-lactamase inhibitors. So these are referred to as the extended spectrum cephalosporins. However, very quickly it was realized that bacteria are now producing extended spectrum beta-lactamases and are targeting even the later generation of the beta-lactam antimicrobial agents. Another thing to worry about is that they're encoded on plasmids. Let me see who's still awake. What are plasmids and how do you think? Why do we worry about plasmids if we are talking about production of beta-lactamases? What are plasmids? Nuclear material in the cytoplasm. Someone else? What are plasmids? So extra chromosomes DNA, extra chromosomal DNA, so small circular double-stranded DNA molecule that is distinct from a cell's chromosomal DNA. Circular double-stranded DNA molecules, genetic material in the cytoplasm. So yes, these are extra chromosomal genetic material. So extra chromosomal DNA molecules that can self-replicate. Yes, for bacterial evolution and adaptation, so very important for that. So a reminder is that bacteria do not have a nucleus because they do not have a nuclear membrane, but they have the chromosomal DNA, which is located in a structure part of the cell that we refer to as a nucleoid. Now, some bacteria, not all, have extra chromosomal DNA, as someone has correctly said. So, have you done bacterial genetics, the lecture on bacterial genetics? Why are plasmids important? Yeah, so circular, so you have not yet done the bacterial genetics. So I'll very quickly, but there's someone here who seems to know. Let me read out what they've said. Plasmids are circular extrachromosomal DNA, and the genes contained in the plasmid can be shared through sex pili in the process of conjugation. Yeah, that's correct. So these are extrachromosomal DNA material. Now remember, DNA carries information. So some plasmids carry different information. You can have plasmids carrying information on how to resist penicillins and so on. And so, yeah, they confer, yes, confer few selected genes like resistance. Yes, that's important. And the worrisome part about plasmids is that they can be shared with other bacteria through a process we refer to as conjugation through the sex pili. Remember again, when you're looking at the bacterial cell, you can have sex pili or the common pili. Common pili are important for attachment of the bacterial cell onto the host cell, which is usually the initial process of infection. And then you have the sex pili, which are involved in conjugation. What is conjugation? It's the exchange of genetic material between bacteria through that process. So you're going to look at it in detail when you look at bacterial cell genetics. And so these plasmids that carry the information on resistance can be shared with other bacteria. And the worrisome part is that it does not have to be a bacterium in the same genus or even in the same species. So for example, a bacterium like E. coli, I'm sure you've all heard of Escherichia coli, can share these plasmids with a bacterium, for example, Neisseria gonorrhea, which causes the STI gonorrhea. So these can be shared between bacteria. It doesn't have to be in the same genus or even the same species through the process of conjugation. Okay. And then you have the class B, which are zinc-dependent metalloenzymes. Now, these have a broad spectrum of activity against all beta-lactam antimicrobial agents. We have the class Cs, which are primarily cephalosporinases, and these are encoded on the bacterial chromosome, which means that they are passed down generations, as opposed to plasmids, which can be shared with different bacteria from different genera and species. Now, these active against the most potent expanded spectrum cephalosporins. Now, you can see there's another term here. So you have the extended spectrum and you have the expanded spectrum. So I'm not sure if you've started looking at cephalosporins in pharmacology. You can familiarize yourself with this. And then you have class D, which are penicillinases, which are found mainly in the gram-negative rods. Remember, rods is another term for bacilli. Okay, so we'll not go into the details of this. So what happened when scientists realized that bacteria are producing beta-lactamases? So there was a need to then inhibit the beta-lactamases, and this is where the beta-lactamase inhibitors came in. So there are two broad classifications of the beta-lactamase inhibitors. So you could have binding of the enzyme. So they form unfavorable steric interactions. And examples are the beta-lactamase inhibitors. Remember I told you for the later generation penicillins and cephalosporins, they carried their own beta-lactamase inhibitors. So here we have the extended spectrum cephalosporins, we have the monobactams, and you have the carbapenems. Then you have the reversible, also known as the suicide inhibitors, very loosely. So these permanently inactivate the beta-lactamase. And examples are clavulanic acid. I'm sure you've all heard of augmentin, which is a brand name for amoxicillin, which is a penicillin, and clavulanic acid. So here you have a penicillin, amoxicillin, then in the same drug, it carries its own beta-lactamase inhibitor, which is no, it doesn't carry its own beta-lactamase inhibitor. It is included in the drug, in the molecule, the beta-lactamase inhibitor. I'm sure you've all heard of augmentin. Another brand name is amoxiclav. Then you have other sulbactam, which tends to be a bit more expensive. So what you mostly find in the market is a clavulanic acid. We also have the tazobactam. Okay. But now what is quickly being realized is that we have bacteria that are inhibitor resistant, or that produce inhibitor resistant beta-lactamases. So the bacteria seems to always be ahead, which is why I think your next lecture will be on antimicrobial stewardship. We must protect the antimicrobial agents that we currently have, because the process of making new antimicrobial agents takes a very long time. So our best defense now is to protect the molecules that we have. So any questions so far? So another broad way of target or mode of action. So we were looking at interference. We are still looking at interference with cell wall synthesis. We have looked at the large group of the beta-lactam antimicrobial agents. And now the other group are the glycopeptides. Now, glycopeptides are large molecules, so large that they are unable to penetrate the outer membrane of the gram-negative bacteria. Remember, we said that the outer membrane of gram-negative bacteria is not freely permeable. Molecules have to pass through the porins to get into the bacterial cell. And so the spectrum of activity for the glycopeptides is limited to gram-positive organisms. How do they act? They form a complex with the D-alanine portion of the cell or precursor. Now, these are reserve drugs. What are reserve drugs? Reserve drugs are drugs that ideally should not even be in circulation in chemists and so on. They have been reserved for specific purposes. So glycopeptides have been reserved as a last resort for multi-drug resistant gram-positive organisms or infections caused by multi-drug resistance or MDR gram-positive organisms. Examples are the methicillin-resistant Staphylococcus aureus. You'll be hearing a lot about MRSA and also the enterococci. So examples of the glycopeptides are vancomycin, teicoplanin, and telavancin. So you'll be hearing a lot when you start practice. Vancomycin is the one which is most readily available. So we are still looking at interference with cell wall synthesis. Another molecule is bacitracin. Again, limited to gram-positive bacteria, but it's too toxic for systemic use. And so we do not use it systemically. It's used in topical preparations. So for example, an ointment, an antibacterial ointment, and so on, but also important in bacteriology because it helps us in the lab to make a presumptive identification of gas. Gas is group A streptococci. group A streptococci. Another antimicrobial agent is cycloserine, which is antibiotic because it's a natural product of bacteria of the genus Streptomyces. It's an analog of D-alanine, so you can see where it acts in cell wall synthesis. Another agent is phosphomycin. Okay. So let me finish this. Someone is asking to repeat glycopeptide mechanism of action. And phosphomycin, so again, this is an antibiotic produced by some bacteria in the genera Streptomycin and Pseudomonas. It targets the Muir enzyme. The Muir enzyme is one of the enzymes that are involved in the process of peptidoglycan synthesis. So the process of peptidoglycan synthesis is very complex. So we've not gone into it. So if you're interested, you can Google that and look at the process. Then we have isoniazid, also interferes with cell wall synthesis. But this is also an example of a reserve drug. So it's a drug that has been reserved specifically to target or for the use of infections caused by Mycobacterium tuberculosis that causes TB. So it forms a complex. Someone was asking about the glycopeptides mechanism of action. It forms a complex with a D-alanyl portion. So this D-alanyl transpeptidation is important because it's what links different chains together and forms a very strong peptidoglycan layer. So once it forms a complex with the D-alanine, you can no longer continue with the interlinkage, which means that you weaken the cell wall. So again, this is an example of an agent which is bactericidal because it will lead to weakening of the cell wall and subsequent lysis because of the high osmotic pressure. So I hope that's clearer. The details are very detailed. I will not go into the details of the exact details, but you can always look. You can always Google and see. So the other target is a disruption of the cell membrane. And this class has two agents. So we have polymyxin B. They're both polymyxins, and we have polymyxin E. The common name for polymyxin is colistin. And again, these are antibiotics derived from the bacterium Bacillus polymyxum. Now they act like cationic detergents. Now let's see if we're still together. What's the general charge of the bacterial cell? Is it positively or negatively charged? Any ideas? So yes, it's negatively charged. So the bacterial cell is generally negatively charged. And that is why you have these antimicrobial agents acting like cationic detergents, which means that they're positively charged. And once you bring a positive charge, then you disrupt the membrane. So colistin and polymyxin B have a limited spectrum of activity. Again, these are reserve drugs, so very toxic. These are reserve drugs, and they're active only against gram-negative rods or bacilli, especially the Pseudomonas species. So they are reserved for the treatment of infections by multi-drug resistant gram-negative rods or bacilli. Pseudomonas species, again, is notorious. This is mostly hospital acquired. So you have a patient who's been in hospital for long, in critical care, and they get an infection from hospital. And these tend to be gram-negative bacilli, E. coli, Pseudomonas, but mostly Pseudomonas. So these tend to be multi-drug resistant against all the antimicrobial agents that have been used against it. And that is why colistin is a reserve drug. So a very toxic drug, but it's your last resort when you have a patient with a multi-drug infection, especially multi-drug resistant bacterial infection. And the commonest genus that you are finding is Pseudomonas. Okay. Now the other mode of action is inhibition of protein synthesis. So again, protein synthesis occurs in the ribosomes. The bacterial ribosome, what's the sedimentation coefficient of the bacterial ribosome? And so you have 70S with 2 subunits. The smaller subunit is 30S, and the larger subunit is 50S. Okay. So the targets, the antimicrobial agents that target protein synthesis can either target the 30S subunit or the 50S subunit. So the ones that target the 30S subunit, we have the aminoglycosides, a very large group of antimicrobial agents. These are just examples. So we have streptomycin, we have neomycin, we have kanamycin, gentamicin, amikacin, and so forth. Then you have the tetracyclines. So tetracycline itself, doxycycline, minocycline, tigecycline. Then you have the 50S subunit. You have the macrolides, you have chloramphenicol, the lincosamides, the oxazolidinones. An example is linezolide. So linezolide is also a reserve drug. So you'll not find it over the counter. In fact, you should not find any antimicrobial agent over the counter. So it's a reserve drug. And you have the streptogramins. The commonest used one is a combination of quinopristine and dafopristine. 50S is the large subunit. So you have a 70S ribosomes, you have the 30S smaller subunit, and the 50S subunit. Now, in as much as you have different targeting, having targets on either the smaller subunit or the larger subunit, the specifications are different. So we say that aminoglycosides and tetracyclines have the target on the 30S, but the specific targets are different. So you do not necessarily need to know the specific targets that may be for a higher level, but you can read around it. So aminoglycosides, and of course it is assumed, you've done in biochemistry, the dogma of molecular biology, from DNA to RNA to proteins. So aminoglycosides block the initiation of translation and cause the misreading of messenger RNA. On the other hand, tetracyclines will block the attachment of the transfer RNA to the ribosome. And on the larger subunits, you have the macrolides, which prevent the continuation of the protein synthesis. You have chloramphenicol, which prevents peptide bonds from being formed. You have the lincosamides, which prevent continuation of protein synthesis. Oxazolidinones, which interfere with initiation of protein synthesis. You have the streptogramins, which interfere with a distinct step of protein synthesis. So it depends on the specifics. So the specifics at this level may not be very important because the details are a bit, but you may go through them when you're looking at pharmacology, but probably not from the molecular biology point of view. For now, know what targets the 30S. You may know some of the details like blockade of transfer RNA, and also the which ones act in the 50S subunit. Now, the other target is nucleic acid synthesis. So interference with nucleic acid synthesis. The first group are the sulfonamides and the diaminopyrimidines. Now, these affect folic acid metabolism. I'm sure from biochemistry, you looked at folic acid metabolism. So sulfonamides are analogs of PABA. What does PABA stand for from biochemistry? Yes, so PABA is para-aminobenzoic acid. So you still remember your biochemistry. So sulfonamides are analogs of PABA. Examples, sulfamethoxazole, sulfadoxine, and sulfadiazine. So these are the sulfur-based drugs. In your practice, you may come across patients who have allergies against sulfur. So these cannot be used in them. Then you have the diaminopyrimidines, which prevent reduction of DHF to THF. What does DHF and THF stand for? Yes, so tetrahydrofolate and dihydrofolate, yeah. So examples of these antimicrobial agents, you have trimethoprim, pyrimethamine, and cycloguanil. Now, what happens when you're using sulfonamides and diaminopyrimidines? Because they're acting, so these are the later steps in the formation of folic acid. So they're acting on the same pathway, so we tend to use them in combination. So common examples you may have heard of SP, sulfadoxine, and pyrimethamine. And what you'll notice with antimicrobial agents that target nucleic acid synthesis is that they are not exclusive to bacteria. They're also used in parasitology. No, I think that you'll do next year or the next academic year. So they're not exclusive to usage. A common brand name is Septrin. So if a patient is allergic to sulfur, now this you will do when you're doing, I don't know if it will be in your 5th or 6th year, when you're doing therapeutics. Now you're looking at the basics. So that you have a patient who's allergic to sulfur. Why are you giving them the sulfur? What are you treating? What else can you use? So if you're treating a bacterial infection, then maybe you can use a penicillin or a cephalosporin. So it really depends on what you're treating. As I said, they're also used in parasitology treatment, especially prophylaxis, treatment against malaria, SP, sulfadoxine, pyrimethamine. So what can you use instead? The guidelines when you're doing therapeutics, the guidelines always have what's your alternative. If you find a patient who is allergic to sulfur, but it depends. So there is no general answer to what is used to treat them instead. It really is very dependent on what you're treating. So still looking at interference with nucleic acid synthesis, we have the quinolones. So these are synthetic chemotherapeutic agents. Again, from biochemistry, remember the two enzymes, you have DNA gyrase and you have topoisomerase. So DNA gyrase A subunit is a primary quinolone target in gram-negative bacteria, while topoisomerase type 4 is a primary target in gram-positive bacteria. Example, nalidixic acid. Nalidixic acid was a very fast agent in this group. So this has a very limited spectrum, active only against gram-negative bacteria. So in practice, you'll find it being used to treat urinary tract infections. The commonest causes of urinary tract infections are gram-negative bacilli. So you'll find in practice, this being used in UTIs. Now, what happened is because it had such a narrow spectrum, then it was improved. And these are the 6 fluoroderivatives, which we refer to as the fluoroquinolones. Examples are ciprofloxacin, ofloxacin, and others. So the fluoroquinolones, you're going to learn more about them in pharmacology. So still looking at interference with nucleic acid synthesis, we have the nitroimidazoles. These are active only against anaerobic. So I hope you remember the lecture on bacterial physiology and certain microaerophilic bacteria. Now again, we see that for nucleic acid synthesis, it's not exclusive to bacteria. So some of these are also used against anaerobic parasites, as you shall learn when you're looking at parasitology. But examples, metronidazole, a common brand name is Flagyl. We have others, tinidazole, onidazole, nimorazole, and so on. So still have others, nitrofurans. An example is nitrofurantoin. We have novobiocin, which acts on the beta subunit of DNA gyrase. Now, this you also come across in bacteriology. It's very toxic for systemic use. So it's used topically, but also important to us in bacteriology because it helps in the identification of species in the genus Staphylococcus, as you shall learn about when you look at the staphylococci. Then we have the rifamycins. The rifamycins inhibit RNA transcription. We have rifampicin, it's in inactive form rifampin, and these are also reserve drugs, so antituberculous drugs. Rifabutin also, it's a reserve drug. It's used to treat infections caused by a species in Mycobacterium, the so-called Mycobacterium avium complex. So these again are reserve drugs. Okay, so any questions? I think so these you'll cover more when you're looking at therapeutics later on in your course, but it's important to mention this. Antimicrobial combination results. So you'll find in some cases you need to combine antimicrobial agents. So what should you look out for? Is what we refer to as synergism. You need to avoid antagonism. So what is synergism? So synergism is the effect of the combination is greater than the sum of the effect of its component. An example is combining a penicillin and an aminoglycoside. So instead of giving very high concentrations of penicillin to the patient, which can cause side effects, you combine it with an aminoglycoside. And you have to be sure about the effect that this is synergistic. They're not going to antagonize their mechanisms of action. So the clinical pharmacists help a lot when you need to do combination of drugs. You do this with the help of clinical pharmacists. They go into the details more of the molecules. What do you want to avoid? Antagonism. So what is antagonism? You can have a combination of antimicrobial agents which are interfering with the activity of the other. Remember, we said antibacterial agents can either be bactericidal, causing the death of the bacterium, or bacteriostatic. So you should not combine a bactericidal agent with a bacteriostatic agent because the bactericidal agent relies on the process of division. Remember, we talked about how the beta lactams work. The cell has to be actively dividing for the beta lactam to attach itself to the penicillin binding protein. So if you give an antimicrobial agent which is stopping the proliferation, then you're giving a penicillin which cannot act because it needs the active division. And so you are exposing the patient to toxic effects, but not helping to clear the infection. Okay. So you can go on to antimicrobial resistance. So what is antimicrobial resistance? So these are some of the headlines that have been found in some of our dailies, and they continue. So alarm as superbug hits British hospital. Super gonorrhea may go global, become untreatable. You can see this was 6 years ago. So has it become global and become untreatable? You see. And then by 2050, superbugs could kill 10 million people a year. So someone wants a break. How many want a break? You want a break, or shall we continue? So the continues have won. So we continue. Yes, I think we continue. So you can see some of the alarming headlines. Superbug in a British hospital, super gonorrhea. And by 2050, how many years away is that? So you'll be in private, in practice. By 2050, superbugs could kill 10 million people a year. How many people have been killed in COVID, or killed by COVID-19 in 2021? Worldwide? So in Kenya, we are still in the 2000s, I think 2000, maybe 800. I haven't looked at the common figures. So you can see the recent figures. So you can see having 10 million people die because of superbugs. So what are these superbugs? What do you think a superbug is? How would you describe a superbug? So 5.6 million worldwide. How would you describe a superbug? What do you think that is? So a superbug, someone thinks a multi-drug resistant bacteria. A superbug, a microbial agent resistant to treatment. So strains of bacteria, viruses, parasites, and fungi that are resistant to most of the antibiotics. Very important term there, most of the antimicrobial agents and other medications commonly used to treat the infections it causes. A bug that has proven resistant to most drugs and has caused fatalities. Yes. Any other ideas? So multi-drug resistant microbe. So what is antimicrobial resistance and why should we worry about it? So we describe antimicrobial resistance, or AMR, when antimicrobial susceptibility. So susceptibility is that ability. No, that's a wrong way to define something. So the antimicrobial susceptibility has been lost to such an extent that the drug is no longer effective for use. Now, when you talk about multi-drug resistant organisms, it's not only one drug. So you're looking at the first line drug, the second line drug, the third line. So the organism is said to have achieved resistance. Okay. So there are different mechanisms of drug resistance, but we divide them into two. We have the inherent or the natural resistance, and you have the acquired resistance. Now we worry more about the acquired resistance because bacteria keep on acquiring the resistance and it continues. And remember, we talked about plasmids. If they get the gene for resistance, they can share it with other bacteria. But what is inherent resistance? So you can have a lack of a transport system for an antimicrobial agent. So even if you give it in an infection being caused by this bacterium, it will not be incorporated into the bacteria. Lack of the target of the antimicrobial molecules. In our introductory lecture on bacterial cell, we see that there is a genus of bacteria that do not have a cell wall, and these are the mycoplasmas. So even if you give any antimicrobial agent that targets the cell wall, it will not be effective, it will be useless. And we already talked about the glycopeptides, where the outer membrane establishes a permeability barrier against the agent. So if you have a molecule that's very large, it will not be able to pass through the porins. Okay. Now, acquired mechanisms of antimicrobial resistance can either be chromosomally mediated, so passed down generations, and we refer to this as mutation. So there can be a mutation in the gene of interest, or they can be plasmid mediated. So remember, plasmids are extra chromosomal genetic material, and this can be shared amongst bacteria through the process of conjugation using sex pili. Okay. But how do you think it comes to this? What are some of the, let's see what's next. What are some of the causes of antimicrobial resistance? How do you think this occurs? How do you think this occurs? So we have multi-drug resistant organisms in hospital setups. So what is causing, what's driving this antimicrobial resistance, do you think? So let's see what you think. Yes, mutations and plasmids, yes. But what's causing the mutation? So evolution of organisms is correct, but what's the process of evolution? So doctors over-prescribing, I don't know, I think over-the-counter medication, not finishing prescribed dose, overuse of antibiotics. But how does this cause mutation? How does that cause mutation? So Hassan Abdallah, selection pressure. Tell us how this selection, what do you mean by selection pressure, and how this will cause antimicrobial resistance? So there's someone here, killing of the normal flora. So Abayesu, your first name is difficult. How do you think this causes antimicrobial resistance? So I think NM, you are replying to the question of selection pressure allows the bacteria to select the genes that work against the antibiotic and pass it on to their progeny. Yes, so that is in mutation. Normal flora, I am following the line of thought of killing of the normal flora. So environmental factors, for example. Drifting and shifting of microbials, what does that mean? So allow the bad bacteria to thrive as the good bacteria are killed. Okay. But what if they are killed? So why is that a problem? So I can see you all have the ideas. Let's see what someone, alteration of metabolic pathways, no defense. Okay, so there'll be no defense. So remember we said at the beginning is that we have not had new antimicrobial agent molecules for almost 25 years. And that's why we need to protect the molecules that we have. So what happens? I wanted the connection between the normal flora and all the factors that you have looked at. Wrong prescription, over-the-counter medication, but how does that then cause antimicrobial resistance? So you have, in Kenya at least, you have someone with a sore throat. So they go to a chemist and they say they have a sore throat and they are given an antibacterial agent. So how does that contribute to the patient who will be in critical care? So long use of same antimicrobial agent. So how does that contribute to the person in critical care who will end up with an with a multi-drug resistant bacterial infection? So the person with a sore throat, now what you need to remember even in your practice is most of these infections are caused by viruses. Most of the infections, common cold, pharyngitis and so on are caused by viruses. So you do not need an antibacterial agent. But most people will not seek medical attention. They'll go over the counter. You're not supposed to sell antimicrobial agents over the counter, but it's done anyway. So that those are policy issues. So they buy the antimicrobial agent and they take it, but they do not have a bacterial infection. But as someone says they had normal flora, I think from your chart, I can see that you've already covered normal flora, the lecture on normal flora, which a large percentage of course are bacterial. So you expose this bacteria, and remember normal flora to you is a pathogen to someone else, or even to yourself. So what is normal flora today could be a pathogen depending on different factors, immune depression and so on. So you have this molecule of antimicrobial agent, which is in your body. So what happens? So let me give an example. Streptococcus pneumoniae causes pneumonia, but it's normal flora in about maybe 20% of the population. So you have exposed these. So the commonest over-the-counter medication are the penicillins, especially amoxicillin and clavulanic acid. So you have exposed these to the molecule. So what happens? Selection pressure always leads to survival for the fittest. And I think someone had already said that so you have, in this case, it will be the strongest survive. So you have various, they have already acquired the genes. Of course, when you expose them to continue concentrations of the molecule, they're of course they need to survive. So they're going to come up with ways of surviving the presence of the antimicrobial molecule. And that's why you now talk about mutations. You have talk of acquisition of plasmids. So the ones without those mechanisms will die. And then you're left with this pool of the superbugs or very fit bacteria. And remember, normal flora is potential pathogen. So the next time these are passed on to a susceptible individual, so it could be from you, a healthcare worker working in a hospital, you do not follow infection control procedures, you infect the patient with these bacteria, which has already acquired the fitness traits of resisting the antimicrobial agents. But it will also be the other way. Healthcare workers can also get this very fit bacteria from their patients. So infection control goes both ways. It's not only for the benefit of the patient, it's also for the benefit of the healthcare workers. So in summary, and in a very simple term, is, as you correctly said, there is a presence, continuous presence of these molecules in our bodies, either through over-the-counter medication. There's a lot of use of antimicrobial agents in chicken food or in cow food or in feeds fed to swine. And there are those animals which were sick, so they have been treated with antimicrobial agents. Remember, the molecules are the same. So amoxicillin in cattle is the same molecule as in animals. And they're supposed to not use those eggs for at least 2 weeks, but these are business people, they need to make money. So you have eggs in circulation that have low concentrations of these antimicrobial agents. And so we are in a state of continuous exposure of bacterial normal flora to concentrations of these molecules. And so they acquire fitness traits. As we say, they need to survive. So we have the mutations, which are then passed on to the progeny or plasmids, which carry the information on resistance. And remember, we said for plasmids, especially very dangerous because they can be passed from this genus to the other genus, from this species to the other species. So to make a very long story short, is that there's a continuous presence of antimicrobial molecules in circulation, which are causing bacteria, the strongest bacteria, which are able to resist the available molecules, antimicrobial molecules that we have, and subsequent, then we get the superbugs. So they're already resistant to the first line, which is amoxicillin. You carry them to hospital where they are, of course, also the other, someone talked about environmental factors, the way we dispose of, what happens when you can no longer use the drug, or when patients do not finish the dose, they throw it in the dustbin. Where does the garbage end up? It ends up in the dump site, it rains, and then it gets into the water system. And so we are in a constant state of concentrations of these antimicrobial molecules, and that is what is driving antimicrobial resistance. So I hope that's a bit clearer, the connection between misuse of antimicrobial agents, selective pressure, selection, and so on. So destruction via incineration, but we have very many clinics that are not what's the word? That are not under the Kenya Medical Board Council. Is it the board or the council? They changed the name. And especially they say that they're not able to police in the, especially the so-called informal settlements. So you have clinics all over the place. How do they dispose of the antimicrobial agents? They put them in the garbage or they throw them, they get to the water system. We know that there's a lot of mixing of sewage with our water in the taps because of leakages. And so that is what is driving this constant supply of low concentrations of antimicrobial agents in our water system, in our foods, in our meat, and so on and so forth. So the ideal is incineration, but that's only ideal. We have very many, an ideal, if there's such a word, of healthcare institutions which are not able to incinerate the waste. Okay, so as we said, for acquired, either chromosomal or plasmid mediated. So what you're going to look here are the results of acquisition of those genes. So the genes can either be acquired through mutation or through conjugation, transfer of plasmids. So what is the end result of having those genes? So one of the most important ones is the drug inactivation or modification. And these are usually plasmid mediated. Remember, we said that they cleave the beta-lactam ring. So we've already talked about production of beta-lactamases. So these act against penicillins and cephalosporins. There are also enzymes that phosphorylate or adenylate, for example, the aminoglycosides against the aminoglycosides. And there can also be addition of an acetyl group, which means that the drug molecule is then inactive. This is common against the chloramphenicol. Now, another result is alteration of the target site. Very common alteration of the penicillin binding protein, which is the binding target site of penicillins. This is seen in methicillin-resistant Staphylococcus aureus. You're going to be hearing a lot about MRSA and also other penicillin-resistant bacteria. You can have mutational changes in 30S ribosomal binding site. And this has been seen resistance of enterococci to streptomycin. For macrolides, methylation of the 23S ribosomal RNA. The 23S is found in the 30S subunit. It's found in the 50S subunit. So blocking of the erythromycin binding. Now again, this is plasmid mediated, which is dangerous because it can be shared with other bacteria. Another result is alteration of the metabolic pathway, common in antimicrobial agents, resistance against antimicrobial agents that target pathways. For example, the folic acid metabolism pathway. So some sulfonamide resistant bacteria do not require PABA. So instead, like mammalian cells, they turn to using preformed folic acid. So even if you target PABA, it does not affect them because they already have the formed folic acid. Then reduced drug accumulation. How does this occur? Can decrease drug permeability. So you can have genes that code for a change in number or the character of the porin channels, so smaller or larger. You can also have an increase in active efflux. So efflux is where the bacterium actively pumps out the drug molecule. This is very common, especially in the gram-negatives, very notorious, Pseudomonas again. You get in the molecule, but you have an efflux pump that pumps out the drug molecule. So there are others which are neither inherent or acquired, can have overproduction of the target enzyme. So even if you target the enzyme, the bacterium has enough to carry out its processes. So sulfonamides and trimethoprim, there are some examples there which you can read later, can also alter the intracellular target enzyme. So resistant against quinolones, bacteria have acquired genes that code for modified DNA gyrase and topoisomerase 4. As for rifampicin, bacteria are able to alter the enzyme DNA-dependent RNA polymerase. Okay, any questions on resistance? Okay, if none, let's look at what do you do in the lab? And this will form part of what you do in your practical sessions. And this is the antimicrobial susceptibility testing. So why do you think we carry out antimicrobial susceptibility testing? Why is it important? Why not just this person has pneumonia, give them amoxicillin? They have gonorrhea, give them ciprofloxacin. Why do we need to carry out antimicrobial susceptibility testing? So any ideas, antimicrobial susceptibility testing? You'll be asking for this a lot in your practice. So to check which antimicrobial agent will kill the bacteria more, which is more effective, to preserve routes that have been established, So yeah, you need to preserve molecules. So you don't use the higher generation. If you can use an amoxicillin, which is cheaper to the patient and even to the healthcare system, you don't have to use the reserve drugs because the reserve drugs tend to be more expensive. Yeah, so to find out the particular antimicrobial agent the bacterium is sensitive to, that is correct. So what is the aim of, so we call it AST in short, what is the aim of antimicrobial susceptibility testing? To select the effective antimicrobial drug and to detect resistance in individual bacterial isolates. So this is for the clinician, of course, is important, and for the patient. Select the most effective antimicrobial drug. So you might find that the most effective is also cheap to the patient and cheap to the healthcare industry as a whole. So if you're running a hospital, you want to use the most affordable agents. Now, this is important for scientists to detect resistance in individual bacterial isolates. So ideally, all hospitals should be having ways where they surveil. It's called surveillance. So you need to do a surveillance. Each year you should have a report on, for example, we had 50 cases of pneumonia caused by Streptococcus pneumoniae. Out of this, because it's been sensitive to amoxicillin for long. So out of these, 50% are showing resistance. So of course, that is just data. Data, you need to compare it with previous data for it to make sense. So if in the previous year you had 40% resistance, and this year you have 50% resistance, then you know that you are headed for trouble. If you have what we refer to as antibiograms, an antibiogram is a booklet which guides clinicians on what to use. When you see the patient, they have pneumonia, what is it likely to be? So which is the best antimicrobial agent before you carry out, because you cannot leave a patient very sick without an antimicrobial agent. You need to use what we refer to as empiric treatment. So empiric treatment is that treatment before you know which is the best antimicrobial agent. So there are several methods of carrying out antimicrobial susceptibility testing, generally divided into two: qualitative methods and quantitative methods. So qualitative methods only answer the question, is it susceptible or is it resistant? So it's either yes or no. So the result looks like, so you isolated this bacterium from the patient's sputum, a patient who had pneumonia. So it's resistant to, the bacterium we have isolated is resistant to amoxicillin, but it's sensitive to which drug? Ceftriaxone. So you can use ceftriaxone. The quantitative methods tell you by how much. So you have a bacterium which is resistant to amoxicillin, but you're interested in knowing by how much. So there will be some numbers involved in the quantitative methods. So qualitative methods, the one which you're most likely to see in your practical is a disc diffusion, also known as a Kabi-Bauer disc diffusion test. So you can use a multi-disc. So these discs contain different antimicrobial agents with different concentrations. You can also use a single disc if you're only interested in finding out, is this bacterium what's the susceptibility of this bacterium that have isolated to, for example, amoxicillin. So this is what the Kabi-Bauer disc diffusion method or the multi-disc diffusion method looks like. So you have a patient, they have pneumonia. You get sputum from the patient, you bring it to the lab, we isolate the bacterium. That's about day 2-3. So in day 3, then you want to find out which is the best antimicrobial agent to use. And so we use a basic agar. I hope you remember the classifications of the agars that we did when we were looking at bacterial physiology. So this is nutrient agar. You can see the disc. So I'm looking at your right. These discs contain different antimicrobial agents, amoxicillin, for example, ceftriaxone, ciprofloxacin, and so on and so forth. So the choice of antimicrobial agent is guided. You learn later how to choose once you do the different bacteria. You're not going to choose vancomycin if you have isolated a gram-negative bacterium. Remember, we said glycopeptides cannot be used in gram-negatives. So you incubate and you come the following day and you're interested in what we refer to as a zone of inhibition. So when you look at the diagram to the left, is the explanation of how we carry out the disc diffusion test. So you have the growth, you have the antimicrobial disc, and you're interested in this zone of inhibition, because what happens is the antimicrobial agent diffuses into the medium and inhibits the growth of the bacteria that you had isolated, that you had inoculated onto this petri dish. So what you need to do is to use a ruler to measure the zone of inhibition. And then we have standards which will tell you if you have 15 millimeters in amoxicillin, then that's resistant, or this is susceptible or something like that. So that's the qualitative disc diffusion method. Then you have the Stokes method also. So for the Stokes method, you have to the right, you can see examples of the Stokes method on the petri dish. The explanation is to the left. So on the same disk, you are divided into two. You're using the same antimicrobial agent as opposed to the multi-disc. And what you have on one side is a control organism. What is a control organism? A control organism is a bacterium whose properties we know and usually get them from manufacturers. So you have the control for example, Streptococcus pneumoniae. So you know this control, the manufacturer will tell you is sensitive to amoxicillin or it's resistant to amoxicillin. And so the test bacterium is what you have isolated from the patient. You do not know its characteristics, and so you're testing it. So you look at the different zones of inhibition and you compare. We don't use it much, but it's important to understand it. For the Stokes method, you can read this later. For the sensitive organisms, the zone of inhibition is equal to or greater than that for the control organism. Then you have the resistant organism, where the zone of inhibition is less than that for the control organism. Okay. Now, nutrient agar is used. There's a note there. If you remember from that lecture, nutrient agar is basic medium. Okay. Antimicrobial combination results. This is where you're looking at what bacteria, what antimicrobial agents can I use? We may not go into this, but this will be shown in the practical. It will be easier to understand. So what you're looking for is the zone of inhibition is bigger when you combine the two as opposed to just one antimicrobial agent. Now, there's some factors that can affect your results that you need to be wary of. The pH of the medium. So when the medium is being prepared, the pH, the components need to be put into account. Is a drug stable at the temperature of incubation? What's the size of the inoculum? We have a standard inoculum for standardization. The length of incubation, is it too short or too long? The metabolic activity of organisms. You can have organisms that are not dividing, so they're not going to give you good results. And of course, the temperature of incubation, it has to be correct. Now for the quantitative methods, we use the dilution methods. The most common one is the tube dilution method in our setup. So that's what you'll see probably see in your practicals. We also have the agar dilution method. So for the quantitative methods, you're looking for two important parameters, MIC and MBC. So MIC is the minimum inhibitory concentration, which is the lowest concentration of antibiotic that inhibits visible growth. And then you have the MBC, which is the minimum bactericidal concentration, which is the lowest concentration of antibiotic that kills about 99.9% of the inoculum. Remember, we said for the quantitative test, you're not only interested in whether there's resistance or susceptibility, it's by how much. Okay, so you add dilutions of antimicrobial agents to a broth or agar medium. Now, this is a standard I was telling you we use, a standardized inoculum of 0.5 McFarland. So this is a tube dilution. And you can see the concentration. So the different tubes have different concentrations from 1 microgram per milliliter all the way to 32 micrograms per milliliter. It depends on what antimicrobial agent you're testing. So you incubate, come the following day. You're interested, remember, in your MIC and in your MBC. So MIC, minimum inhibitory concentration. Remember also in that lecture on bacterial physiology, we're looking at the broth. The broth is a liquid medium. How do you tell there's growth in a broth? There will be turbulence. So the last tube without turbulence, that will be your MIC, 4 micrograms per milliliter. It's usually very tedious because you've not yet finished. You need to get your MBC. So you do what we refer to as subculture on solid medium, and then see if there's any growth. So for the one with 8 micrograms per milliliter, there is some growth, which means this cannot be the MBC because you have not killed all the bacteria. And so the first tube that gives you no growth is what we refer to as the MBC. So in this case, the MBC is 16 micrograms per milliliter, and the MIC is 8 micrograms per milliliter because it was the last tube that did not show any growth. There was no turbulence. However, in as much as there was no turbulence, there was still some live bacteria, which means that it was not able to kill all the bacteria. So that's your MBC. Tube dilution is usually difficult to understand at the first explanation. It becomes clearer during the practical. When you look at this and it's explained practically. It's very tedious, but very important because you can have bacteria that are not showing any resistance when you do the multi-disc diffusion. But when you look at the dilution, you can see that there's the concentrations are rising. For example, if last year, the MIC of Streptococcus pneumoniae to amoxicillin was 8, then this year you're finding 16. So you can see that there's an increase in, and you're moving towards resistance. So this becomes clearer when you go for your practicals. So make sure you understand this during your practicals. Now, the other method is e-test. E-test is actually a hybrid method. You're a hybrid of both qualitative and quantitative. So it uses a plastic strip impregnated with antimicrobial agent. It's placed onto an agar which has already been inoculated with bacteria. And there's diffusion of the antimicrobial agent into the agar. And the e-test usually has a concentration gradient as shown here. So you have a strip TM could be trimethoprim. So you have an IP could be imipenem. So you have a strip, you can see the concentrations, and you can see the diagram showing MIC. So you can read the MIC directly from the strip as opposed to using the very tedious tube dilution test. And you have the automated AST. Automated to a point, it's not that you get a sample from a client and you put it into the machine. It doesn't work like that. You still have to go through isolation of the bacterium, what we refer to as the purification of the culture, and then doing the standardization, which is a 0.5 McFarland, and then putting it into the machine. So it's not as automated as it sounds. There's still a lot of manual work to be done. Finally, you have the mechanism-specific tests. We looked at some of the mechanisms. The most important is the beta-lactamase production. So what is used is what we refer to as a nitrosamine disc. Nitrosamine is a chromogenic cephalosporin. What does that mean? When its beta-lactam ring is cleaved, it changes color. So when you have a patient with, for example, pneumonia, you have isolated Streptococcus pneumoniae, you very quickly need to tell the clinician, can you use the beta-lactams or not, as you get the antimicrobial susceptibility tests. So you can use nitrosamine, you place it into the petri dish that contains your isolate from the patient. So if it changes color to red, then you know that it produces beta-lactamase. So you'll advise them avoid beta-lactamase producers, beta-lactams. And if you have to, you need to use a beta-lactamase inhibitor. Okay, so that's a mechanism, an example of a mechanism-specific test. So this is a summary. We have looked at the classification of the antibacterial agents from the point of view of bacteriology. We have looked at antimicrobial resistance, and we have looked at antimicrobial susceptibility testing. So any questions? So someone is asking, the N is used in all four. Nutrient agar, what do you mean by all four? I think you asked a bit earlier. So nutrient agar is a basic medium. Why do you use a basic medium? Because you do not want anything affecting your results. So we mostly use nutrient agar. However, there are some fastidious organisms in which you need to use a special medium for antimicrobial susceptibility testing. So it's not always used, but nutrient agar is the most commonly used. So someone is asking, how did the UK overcome the MRSA outbreak of 2004-5? So I think you've read about it. Do you have any idea what they did? MRSA. So because of time, you can go and read about this and other outbreaks. What did they do? There have been outbreaks of MRSA in NICUs, that's the neonatal ICU, HDUs of the multi-drug resistant bacteria. So we've not yet done the individual bacteria, so I'm careful in mentioning some of them, but read about MRSA outbreaks. What was the other latest outbreak? We also have some fungi that are showing multi-drug resistance. Pseudomonas has caused quite a number of outbreaks, in which case you use colistin, but the worst part is that you almost always suffer loss of life because of these multi-drug resistant organisms. So for bacteria that gain full resistance, so I'm assuming you're talking about multi-drug resistance. So as you look at the individual bacteria, there are two terms that you'll be coming across: multi-drug resistance and extensively drug resistance. So if I can give an example of TB. So TB, we have first-line treatment, which goes for about 6 months. And then we have multi-drug resistant TB, MDR TB. So for MDR TB, you cannot use the first line, you need to use the second line. But then we have those strains of Mycobacterium tuberculosis, which are referred to as extensively drug resistant, which means that you have to use a third line. And then there are those that cannot, even with the third line, because the third line is not yet standardized. It depends on the country. So there are those, and that's why the danger with antimicrobial resistance, because we do not yet have, we don't know what to do. As I said, we do not have any new drug molecule for the last, I think, 25 years. So if you have an extensively drug-resistant bacterium, all you can do is to, most of the time, you're fighting the symptoms. You're unable to control and most of them tend to be, not all, most, especially in critical care, tend to be overwhelming gram-negative sepsis. So they actually die from this because you do not have the drugs or they're not working. And then you get super infections from other bacteria and so on and so forth. So how do you help? Sometimes you really, it's just support. It's the support of the patient. Sometimes you're not able to do anything if you do not have, if you get to third line, by the time you're talking about colistin for overwhelming gram-negative sepsis, really it's at the end of life. So if it doesn't work, and it's also very toxic, so you cannot use it for a long time. So either the patient dies from the sepsis or they die from the toxicity. It's a very delicate balancing act, which is why I think in your next lecture, you're going to talk about antimicrobial steward. How can you protect these molecules, drug molecules that we have, so that you do not get into a state of extensively drug-resistant bacterial infections. So it's not, Noella, it's not a general answer. It's usually very patient-specific. What does this patient have? Which infection is that? What drugs are working? So what's the next line? Sometimes you do not have a next line, and really you just wait for, you do symptomatic management, you do the best you can. But in the end, you probably lose them because of the sepsis. So any other questions? So if not, you can call it a day. Thank you. I'm sure the class rep will have the recording.